In today's lecture, we're going to review DNA structure and also replication of DNA. DNA is made up of nucleotides, and there's 4 different types of nucleotides. So all of this should be pretty much a review, but just to remind you, once again, that a nucleotide is comprised of sugar group, a covalently based group, which contains nitrogen, carbon, in either a single or double ring structure, and also phosphate group. The base group varies in such that there's 4 different bases: adenine, guanine, cytosine, and thymine, and the one that's shown up above here happens to be adenine. Remember that these nucleotides come together to form a single-stranded molecule, and they link between their phosphate groups and the hydroxyl groups of the sugar of a sugar group. This is called a phosphodiester bond. And a single-stranded is associated also with another strand to create the double-stranded DNA molecule. And those 2 strands run in anti-parallel direction, which you can see in this particular scenario here. The 2 strands are held together by hydrogen bonds between the bases. G always pairs with C, and 3 hydrogen bonds hold that base pair together, and A always pairs with T, and there are 2 hydrogen bonds that hold that base pair together. The double helix, or that 2-stranded molecule that is DNA, twists in a helical nature, and essentially when it twists, you can see that it creates smaller grooves and also larger grooves, which are called minor and major grooves. So the twist causes this different twisted nature as far as the spacing. Now keep in mind, when we think about deducing the structure of DNA, there were several different scientists that were involved in this process. The different types of experiments that really identified DNA as the genetic or heritable material, where the first studies were done in the early 1900s, and it wasn't until 1953 that Watson and Crick revealed the structure of DNA, consisting of nucleotides in this 2-stranded double helical nature. But many other scientists besides Watson and Crick contributed to the ability of those 2 people to actually determine the molecular structure of DNA. When we think about the significance or importance of DNA, remember that essentially it stores the genetic information. Essentially, when we think about genetic information containing information about how to make the individual proteins of a cell, it also has to store the information on how to replicate itself, and also information on how to express the genetic material, essentially how to lead to protein production. So this particular scenario is just defining the fact that we have DNA. DNA has specific genes on the DNA that specify for particular production of certain proteins that these proteins lead to specific genetic traits. This DNA that contains specific genes and other material essentially needs to be copied and duplicated, so we pass it on to new cells. So in the important process of DNA is it obviously is the genetic material, but it has to be able to be passed on. So all cells have genetic material, and also the information needs to be accessed in such a way that the protein building instructions are ultimately going to lead to production of proteins. So there needs to be important information on how to express certain genes. When looking at DNA between prokaryotes and eukaryotes, there's some big similarities. They all are comprised, regardless of prokaryotes or eukaryotes, comprised of nucleotides and have the same types of structure within their nucleotides. So deoxyribose sugar, phosphate groups, and the 4 different bases. And the assembly into that single-stranded molecule associated with another stranded molecule forming duplex structure is similar. However, prokaryotes have a single chromosome. In this particular figure, the single chromosome is highly supercoiled. Prokaryotes also can carry different types of accessory DNA and what are called smaller circular plasmids. But in general, when we think about prokaryotes, they have a single chromosome. And eukaryotes, on the other hand, have a larger genome In fact, something like yeast cells contain 1.35 times 10 to the 7th base pairs, so their amount of genetic material or sequencing is much bigger. And these chromosomes are then divided up into separate linear pieces, or the DNA itself is separated into separate linear pieces, which we call chromosomes. And different organisms, different types of eukaryotic organisms, have distinct numbers of chromosomes. Humans, for example, have 23 pairs or 46 chromosomes. These linear pieces have differences as well to that single circular chromosome of prokaryotes. They also are associated with histones for allowing high condensing or wrapping of the DNA, and to this date, prokaryotes have not it has not been identified whether prokaryotes actually have histones. But eukaryotes do have histones that are important in the wrapping of DNA and dealing with the massive amount of genetic information. Specifically important when we think about condensation of chromosomes for proper segregation during division. So this bottom table just highlights some of those important differences. Another important difference between prokaryotes and eukaryotes in regards to structure is the fact that the circular prokaryotic DNA is largely comprised of sequences that are important for protein production, whereas you'll see now, in light of much of the genome sequencing information, that eukaryotic DNA has only a small fraction that's actually DNA that's specifically important for production of proteins, and it has high levels of what's called repetitive non-coding sequences. Also, DNA is stored in different locations. You know that basically prokaryotes lack a nucleus, so there is no specialized compartment that we see in eukaryotes, which is the nucleus. And this allows for some important distinctions between DNA in both prokaryotes and eukaryotes. Now, the human chromosomes, or what's called the human genome, was preliminary sequenced in the early 2000s, so around 2000, by two different companies. And now later from that, 2004, this genome sequencing project was actually finished. And they found some important things in relationship to eukaryotes as well as humans. The human genome, as you can see after sequencing our 46 different chromosomes, contains about 20-something thousand genes. At the time, this was actually a much lower number than they predicted based on the variety of proteins that comprise our cells. But what they did notice is essentially these protein coding genes have non-coding regions in them called introns, and that allows for alternate splicing of mRNA to account for a higher number of proteins from a single gene. Additionally, allowing for a greater complexity of proteins as well is the fact that we have a variety of post-translational modifications that can alter function and activity of proteins as well. So even though we have a fewer number of genes that was predicted based on our complexity, ultimately the ability to alternately splice mRNA and different post-translational modifications can increase the number and functionality of different proteins in something like humans. If we look at the estimated genes or protein coding genes in different organisms, and you can see in blue, that is the actual protein coding genes, and you can see that humans, here's our approximately 20,000 protein coding genes, but you can also see many other eukaryotes have close to the same number of protein coding genes as we do. Yeast has a few less, but you look at something like a sponge, sea anemone, etc., and essentially they have up to, close to 20,000 different genes. But you also will notice some other types of eukaryotes that have a much greater number of genes, and oftentimes a greater number in genes might not reflect an increase in complexity of the organism. Sometimes we get an increased number in genes in certain types of organisms due to duplication, etc., but it necessarily may not increase the actual number of proteins that that particular organism has. When we think about kind of what they found from looking at the different genome sequences of different organisms, what they found from eukaryotic genomes is the fact that we have a high amount of repetitive DNA sequences. So this is something you don't see in prokaryotes. What are the different types of repetitive DNA? There's what's called satellite DNA, which is the centromeric regions, which is going to be important for connecting sister chromatids together. And there are also what are called mini and micro DNA, which are short repeats of similar sequence repeatedly. There's also moderately repeated DNA sequences. These are important for ribosomal and transfer RNAs, histone proteins, and various sequences with non-coding functions as well that are considered moderately repeated DNA sequences. And then obviously there's a small portion that we know is non-repeated DNA sequences, and these are the portion that are actually protein coding genes. So there's a very small portion of actually protein coding genes with a high amount of repeated DNA sequences throughout the eukaryotic genomes. Now, when we think about replication of DNA in general, this kind of highlights some key difference just simply thinking about prokaryotes and eukaryotes. And then once I describe this figure, we're then going to go in and talk about DNA replication in prokaryotes, and then talk about some key differences in complexity in eukaryotes. Now, keep in mind, prokaryotic cells have a single chromosome, which is circular in nature, and ultimately that circular chromosome will replicate and then separate, and you're left with two chromosomes that will get divided into two new prokaryotic cells. Keep in mind, in eukaryotes, we have multiple chromosomes, and these chromosomes, remember, are associated with histone proteins, which assist in wrapping, which are these dots that you're seeing along this chromosome. The ends of chromosomes have what are called telomere regions, and ultimately the size of these chromosomes are much larger, and we have multiple chromosomes to actually replicate in the replication process, which makes it different than the replication process in prokaryotes. When we think about the simplicity of DNA replication in bacteria, the replication is going to start at a single origin. There's a special sequence that's called the ORIC, and this is where the replication machinery will assemble, and ultimately replication will start at that site. The replication will then move bidirectionally in opposite directions, which you can see, until the chromosome has been effectively copied, and then the two chromosomes will be separated following replication for division of the two cells. When we think about, in general, this replication process, replication occurs at what are called replication forks. So this is where the DNA is essentially going to be opened up and essentially being copied. That's a replication fork, and it moves bidirectionally at these replication forks. When synthesizing new strands in prokaryotes and also eukaryotes, keep in mind that this process is what's called semi-conservative replication, and that is because the two strands will pull apart in the DNA molecule, and each will serve as a template to build a new strand on top of the old, which means semi-conserved. The new DNA molecule, when it's completed, will be half old and half new, which is where it gets the term semi-conservative replication. And there were several different experiments that highlighted the fact that DNA was in fact replicated in what's called a semi-conservative fashion. The DNA replication occurs in such a way that new nucleotides are added to the 3' hydroxyl group of a sugar, and a new base is added to that sugar of a base above it through interaction or joining of the phosphate group to that sugar group. So you get joining or linkage between hydroxyl groups of sugars to phosphate groups to hydroxyl links of sugars to phosphate groups, and this creates what's called the backbone of the DNA strand. And the base groups will sit out in the middle, which you can see here, for hydrogen bonding to hold the two strands together. So this particular scenario is actually showing you this base being added where ultimately a phosphodiester bond is being generated due to linkage of that hydroxyl group to that phosphate group. And you lose 2 of your phosphate groups in this process, which you can see here. There's special metal islands that are thought to be important in helping catalyze this reaction and stabilize the pyrophosphate during its release. This particular diagram here is showing you a replication fork where the 2 strands are being pulled apart, and ultimately replication or synthesis of the 2 new strands. And we're going to talk a little bit more about replication and how this occurs, how both strands are effectively replicated. Now, when we think about replication in general, at these replication forks, what has been discovered is one strand is placed in a continuous fashion, and this is called the leading strand, and it moves towards the replication fork. It's in the same direction as the replication fork. So here's the replication fork. The other strand is laid down in fragments or pieces. These fragments or pieces are called Okazaki fragments, which was the name of the scientist who first discovered the segmentation of the strand. And this strand that's laid down in segments that actually are laid down in opposite direction as the direction of the replication fork is called the lagging strand. And regardless of prokaryotes or eukaryotes, replication occurs in such a way that there's always a leading strand and lagging strands. Now, there are several different players that have been important in the replication process, that have been discovered as being important players in the replication process. Some of the first discoveries, of course, involved prokaryotic replication because it's a little bit more simplified. And ultimately, different types of mutagenesis studies as well as reconstitution in vitro experiments has really provided insight as to what important players are involved in the replication process. And as scientists have moved into eukaryotic systems, you'll see that many of these players are also involved. Similar types of players are also involved in the eukaryotic replication process. There just seems to be more complexity and essentially more players involved. So let's look at a few of these different proteins that have been discovered that are important for replication of DNA. And a few of them are shown here. Helicase, primase, single-stranded proteins. So let's kind of go through a few of these so that you can understand some of the different roles of these different players during DNA replication. Helicase, which you can see here in pink, helicase is important for breaking the hydrogen bonds in the double-stranded molecules such that you can form the replication fork. So it uses ATP hydrolysis energy to essentially break the 2 strands so that essentially you can reveal the template strand to build the new strands upon those template strands. Single-stranded DNA binding proteins are important because they are essentially going to bind to the single strands once they are broken to prevent them from reforming back into a duplex. Primase, as you're going to see here, primase is important because it actually lays down what's called an RNA primer, which allows for synthesis of the lagging strands. And we'll talk a little bit more in the future about why a primer is important. But ultimately, primase is an RNA polymerase that can lay down RNA that base pairs with a short sequence of the DNA strand, such that synthesis can occur off that primer region. Now, the primase and the helicase together form what's called a primosome, and essentially they're going to processively move along the lagging strand to allow for, first of all, the strands to separate, as well as lay down the primers for each of the Okazaki fragments. The replisome is the whole machinery that's going to be involved in DNA replication. So this is often called the replisome. So a replisome is all the important components that are critical for DNA replication. And that includes DNA helicase, primase, single-stranded binding proteins, but it also includes DNA polymerase and other proteins as well. So ultimately, this is called the replisome, kind of involves all the important components involved in DNA replication that's going to lead to production of both the lagging and the leading strands of the DNA. So one of the most important players is obviously DNA polymerase, and the role of DNA polymerase is the fact that it's going to be involved in synthesizing the new DNA. These new strands being made by DNA polymerase, there's some important steps required for this process. So after looking at the activity of DNA polymerase and looking at what might be required or not for its activity, they discovered the fact that there needs to be a primer for DNA polymerase to synthesize the new strand. It cannot start a new strand of DNA. It actually has to start off a pre-existing strand, in this case a primer. There has to be a 3' hydroxyl terminus for DNA polymerase to start a phosphodiester bond of the newly synthesized strand. None of the 3 DNA polymerases that have been discovered in prokaryotes can initiate DNA chains, and therefore a primer is required, which requires what's called primase, which is an RNA polymerase. This is also true for eukaryotic polymerases as well. After analyzing and discovering polymerases in E. coli, they discovered that there's actually 3 different types in E. coli, which seems to be true for most prokaryotes, and in fact polymerase 1 was the first discovered. However, the DNA polymerase 3 is the polymerase that's important for replication of the leading and the lagging strands, so replication of the new DNA. Pol2 activity is not required for DNA replication, and Pol1, DNA polymerase 1, appears mostly active in DNA repair. Ultimately, when we think about how replication occurs and looking at both the lagging and the lagging strands are also what are called Okazaki fragments. The important players you can see here, this is the replication fork, and here's our leading strand, and here's our lagging strand. You can see the leading strand is made in the direction of the replication fork, and the lagging strands are actually made in the opposite direction as the replication fork. In this direction, in this direction, and they're laid in pieces towards the replication fork, but the direction in which they're synthesized is away from the replication fork. Now, you'll notice the primers that are ultimately required for each of the Okazaki fragments so that DNA polymerase can assemble off of those primers and continue the strand. And the primase is going to be obviously important for assembly of those RNA primers that the DNA polymerase will then assemble off of. Now, you'll notice the directionality, and DNA polymerase, as well as its other important features, it can only assemble a new strand in what's called the 5' to 3' direction. It uses the 3' to 5' template of DNA, but it assembles in the 5' to 3' direction. That is why the Okazaki fragments are laid away from the replication fork, because that is the actual direction of the 5' 3' direction of that side of the 5' to 3' DNA strand. So that has to move in that particular direction due to the 5' to 3' direction. So ultimately, that's why we always end up with a leading strand and a lagging strand, due to that 5' to 3' assembly process. Now, once the DNA polymerase reaches the end of the Okazaki fragment that was completed prior to that segment, then the RNA primers will have to be removed by polymerase 1, and then DNA will then be added back into those regions, and the nucleotides will be ligated together. So these primers will be removed, and DNA deoxyribonucleosides will then be added, and the RNA will be removed, and the pieces ligated back together to create continuous strands, so ultimately to seal these fragments together. Another important player, besides thinking about DNA polymerase, primase, and also helicase, there have also been what are called topoisomerases that have been discovered, and topoisomerases are supposed to help alleviate some of the supercoiling that occurs as you pull two strands apart. So if you can envision here on this side, if you had two ropes that are twisted together and you try to pull them apart, you'll get tightening of the helical structure above it. And this is exactly what happens during DNA replication. Is as you pull the two strands apart, you break, as the helicase breaks the hydrogen bonds, it actually causes the upstream DNA to supercoil. This creates a lot of tension on the DNA, and it makes it even harder to pull the two strands apart. So there's special proteins called topoisomerases that have been discovered. In prokaryotes, it's called gyrase, and there's what are called topoisomerase 1 and 2, which have special names as well in eukaryotes. So there's two different classes in eukaryotes, and the two different types, 1 and 2, are different based on how they break the hydrogen bonds. One can cause a single break, and the other can break both strands, and they help reanneal the DNA minus the coil structure. So they help break and then rejoin the strands ahead to relieve the tension due to that supercoiling, due to the pulling apart of the two strands. So DNA polymerase III, just as a summary to remind you of kind of its important properties, one of which it has multiple subunits in prokaryotes, and these different subunits provide some different functions, and we're going to talk about a couple of them, one of them being a clamp-like structure and also what's called a clamp loading complex. The DNA polymerase III in prokaryotes replicates both lagging and leading strands of the DNA, and when replicating the lagging strand, the DNA polymerase III that replicates the lagging strand in prokaryotes actually hitches a ride with the DNA polymerase that replicates the leading strand. So they form a complex together, and they hitch a ride together to replicate these strands. And we'll see that in just a second. One of the important subunits forms what's called the beta clamp, and this is an important structure that allows DNA polymerase to actually stay on the strand of DNA and help slide across it as it's replicating the DNA. And eukaryotes have also been discovered to have a homologue of the beta clamp. So this is looking at the steps of replication and the involvement of these different types of proteins. So if we look in the first figure A, you can see DNA polymerase III. You can see that there's two of them. One on the lagging strand, one on the leading strand. You can see that they're linked together, so they hitch a ride together. And you can see the DNA polymerase on the leading strand forming a continuously newly replicated DNA single-stranded molecule on the template, which the new one is shown in red and the original template is shown in blue. You can see it's replicating in the 5' to 3 direction towards the replication fork, and that there are single-stranded binding proteins to help hold those two strands apart. And helicase is important above or at the start of the replication fork. That's going to be important in pulling those two strands apart. Now, to allow for DNA replication of both the lagging and leading strand by these two DNA polymerases that form together, that they go in the same direction, the only way to do that is if the DNA is actually looped. And you can see that. So the DNA polymerase III that's involved in replicating the lagging strand is looped so that it puts the direction of replication in the same direction as the leading strand by the loop. And you can see now it's in the same direction. It's still assembling in what's called the 5' to 3', but this looping allows these two replication polymerases to link together and work towards that replication fork, and that looping is important for that process. And then once that DNA polymerase 3 that's on the lagging strand reaches the fragment that's already been done, then the loop will let go, and then it will form a new loop to complete the next segment. So you can see that in this next particular scenario here, where it let loose that particular loop because replication was complete on that particular fragment, and then it will move and loop the next fragment in such a way that it will then build that new strand, and the looping will allow for the correct orientation or the same directional production as that leading strand DNA polymerase. And you can see that in this particular scenario now. So ultimately, these two DNA polymerases work together as a single complex to ultimately replicate both the leading and lagging strand. The clamp loader, which you can see, the clamp and clamp loader are found here as part of the subunits of each of these DNA polymerases, and that important beta clamp subunit is that circle feature at the end of DNA polymerase. So this gives you a larger drawing of the replication fork of a prokaryotic chromosome and some of the important components of this holoenzyme. And DNA holoenzyme refers to, DNA polymerase holoenzyme refers to all of its important subunits and DNA polymerase. So here's some of the important subunits. You can see there's what's called a clamp loader. This is an important subunit that actually helps link the ring structure onto the core polymerase so that it will remain attached to the single-stranded DNA and also allow for it to slide across. It looks kind of like a donut with a donut hole. And you'll see that both of the DNA polymerase have this beta clamp, and this clamp loader is going to be essential to helping load that clamp on and reload it, especially when we think about the lagging strand. In every fragment, it's going to have to be reloaded on a new clamp onto that DNA polymerase that's synthesizing the lagging strands. So this replisome, remember, when we think about replisome at the replication fork is all the machinery needed for DNA replication. And this includes the holoenzyme, which is all the important subunits of DNA polymerase, but also other important players such as the helicase, single-stranded binding proteins, as well as primase, which is going to help lay down the primers, the RNA primers to initiate synthesis at the lagging strands. So how does this compare with eukaryotes? There's been some important differences discovered in eukaryotes. The first differences that we can kind of think about in general is the fact that we have a much larger amount of DNA to replicate. And what has been discovered when looking at something as simple as even yeast DNA replication is they've found that replication doesn't just start at one initial replication or origin site, it starts at multiple origin sites. This was originally shown in yeast, and this particular experiment that you're seeing above was due to the addition of radio-labeled nucleotides and DNA, and after a brief period of time, they stopped the replication process and looked to see that they saw these fragments of new DNA being made. And the new DNA was the radio-labeled nucleotides. They saw these fragments. These fragments would represent the multiple origins or replication bubbles that form on the same DNA molecule. So essentially you're getting multiple replication sites occurring simultaneously, so it allows DNA replication to occur at a much faster rate. Now, when we think about replication and something like multiple origins, what you're going to find out is that yeast has special sequences that drive recognition of these certain origin sites, and eukaryotes also have specific regions that are important for starting of replication, but they don't have necessarily the same type of sequences as yeast do. So after looking at eukaryotic cells and their replication, there's still there is a lot that's not well understood. And there's some advances in our understanding have been done using mutant yeast cells, looking for specific mutational changes that prevent replications to highlight specific proteins that are involved in the process, and also in vitro systems such as using Xenopus egg oocytes and taking these extracts that have machinery, all the different important proteins that are needed for DNA replication, and adding or removing certain components and looking to see what would be important for replication as a whole. So some of these in vitro systems have also been extremely effective in helping decipher the DNA replication process in eukaryotes. But there's much to still learn in regards to DNA replication in eukaryotes. Initiation of replication of eukaryotic cells requires replicate at multiple origins, which we just talked about. In small portions, these origins are also called replicons. And initiation of this replicon is regulated. There's specific proteins that help mark origin sites and help initiate the process at each one of those origins. So now let's look at a specific example of origin and recognition of an origin, and also the starting of the DNA replication process in something like yeast. And yeast, remember how I told you there's specific sequences called ARSs that are about 11 base pairs. They look like they're rich in AT bases, and those serve as marking sites for binding of specific proteins that will serve as start sites for DNA replication. One particular important complex that binds to these in yeast are called the origin recognition complex, which is abbreviated ORC. Some other proteins are important in binding to the ORC, such as the MCM2, MCM7 protein, and also CDC6 and CDT1. And ultimately, changes to this complex at the origin, which is initiated by important kinases involved in initiation of the division process, will help lead to opening of those two strands so that we form bubble formation and initiating the replication process. So keep in mind, there are important proteins that mark these sites and also help initiate replication, and the initiation is controlled by multiple proteins, and it's a very elaborate control mechanism, so we ensure we only replicate DNA upon need. After analysis of origins of replication, they now think that mammalian cells have approximately 10,000 to 100,000 origin sites, and depending on cell type and type of DNA, whether it's highly condensed, etc., will depend on how many origins are ultimately used to replicate certain types of chromosomes. But ultimately, there's multiple origins so that replication can occur simultaneously on a single DNA molecule so that it speeds up the replication process. So how does the replication fork vary from something that we saw in prokaryotes? There's many similarities in the replication fork and also machinery that's found at these replication forks. There's been homologs for the helicase that's found in eukaryotes that works in a similar fashion and also has a similar structure. You're going to learn of what's called the CMG complex in your paper that's part of your supplemental reading for today's lecture. So you're going to read a little bit more about a helicase or eukaryotic helicase. So there's helicases that are going to help pull apart the DNA strands. There are also primases and also DNA polymerases, which you're going to see. You're also going to see that there's multiple DNA polymerases and they have different roles, so the complexity increases when we think about DNA polymerases in eukaryotes. There's also important, there's a homolog that's similar to the beta clamp in prokaryotes called PCNA, and there's a clamp loader or similar function to the clamp loader, which is called the RFC, which has been found in eukaryotes. So many of these types of machineries have been conserved, or types of proteins that are involved in this machinery have been conserved and are still utilized in eukaryotes. There's just an increased layer of complexity. Obviously, of course, there's topoisomerases, which are going to be important in relieving that supercoiling due to the breakage of the two strands, and also single-stranded DNA molecules that are going to help hold the two strands apart. So just as a summary, at the eukaryotic replication fork, there's many similarities between prokaryotes and eukaryotes, and there are several DNA polymerases in eukaryotes, so there's more than prokaryotes. And also, these polymerases work in a similar fashion. They only elongate from the 5' to 3' direction. They also require a primer, which we saw before with prokaryotic DNA polymerases. But some, however, have a 3' to 5' exonuclease activity for ultimately removal of RNA primers and allowing for new DNA and ligation of those primer regions, as well as involved in DNA repair. So what do we know in regards to comparison between some of these proteins involved? And you can see here are proteins that are important in E. coli. So ultimately, DNAA is a recognition origin replication protein. We also have recognition of origin proteins. We call these ORC proteins. Both of them contain enzymes that are important for dealing with supercoiling, such as topoisomerase 1 and 2 in eukaryotes, gyrase in E. coli. And there are also other important proteins involved in unwinding the DNA, such as DNA helicases, DNA B and MCM, and also loads helicase onto DNA, which you can see. So there's important proteins involved in that. Single-stranded binding proteins, clamp loader, which is called the gamma complex in E. coli, but the equivalent in eukaryotes is RFC. And there's also some DNA polymerases that have been discovered. In prokaryotes, DNA polymerase III produces both the leading and lagging DNA strand. However, in eukaryotes, you're going to see that there's two different polymerases. There is the delta and the epsilon DNA polymerases that are going to be important in replicating those two strands. There's also an equivalent clamp in eukaryotes, which is called the pCNA. There's been a primase that was discovered in both, which is an RNA polymerase, etc., etc. So essentially, we have very similar proteins that work in similar fashions. However, you're going to see that eukaryotes have a little bit increase in their level of complexity when it comes to DNA replication. Just to remind you that DNA polymerase delta will replicate the lagging strand, and DNA polymerase epsilon in eukaryotes replicates the leading strand. So you will see that also in the paper, the supplemental reading paper. So the supplemental reading paper is looking at recruitment of DNA polymerase epsilon by components involved in also the helicase. So it will allow for a direct recruitment of DNA polymerase epsilon to the replication fork due to binding of a helicase. And so you're going to read through that particular paper, and there's some questions already posted on that. So we do see a difference in regards to what DNA polymerase are important in that replication process. And there's been 5 different DNA polymerases discovered in eukaryotes, and the 2 that are important for replication of the new strands are DNA polymerase delta and DNA polymerase epsilon.